Good morning. All of you have done the attendance? Okay. All right then. I'm Dr. Deeba and I'm from Department of Medical Biochemistry, and I will be teaching you the third portion under the course of General Biology. Now, when you started the course, you started it off with cell biology, which was given by Dr. Sama, and majorly she talked about structural components of the cell as a whole of the tissue, correct? Maybe something functional related to transportation. Then you had Dr. Mahmood, who taught you molecular biology, a little about, how the DNA and RNA and the proteins are made. Now, what I'm going to teach you is to build up on what Dr. Sama has given you. So this is one level up on cell biology. And I will be with you for roughly 3 weeks. And the following are the topics that I will be covering with you, starting on with protein trafficking or protein transport. All of you know what is the organelle. It's ribosomes that make proteins, but how they are transported around the cell or outside the cell, what's the process, we'll learn about that. Then there is protein degradation. Lots of proteins are made in our cells on on the basis of hours, and some are made for years, but eventually they become older, they break down. How do they break down? Then comes the cell biology. Cell biology is or the cell cycle is related to how the cells that are present in our body, how they go through different phases. What are those? And since cell cycle is an important component to maintain the health of the cells, we will talk about the regulation also of the cell cycle. And then will come signal transduction. Now you have learned about receptors, and you know something comes and binds on the receptor, and that kind of acts as a stimulus to the cell. But with me, we will discuss how that signal gets processed inside the cell. Okay? Finally, we'll have cell death, that what are the conditions and how different cells undergo death and why. Ending up with aging. All of us are born, we grow up, and then eventually we start aging, right? But the aging that you see on people starts way before at the cellular level. So what is it? How does it start? We will cover it in under aging or cellular senescence. So this is the overall view of what I'm going to teach you. Now, before I start, a few guidelines regarding how my sessions go by and some of the frequently asked questions. The most frequently asked question is, Doctor, are we only responsible for the slides, or we should go through the book also? This doctor tells you that for her portion and only for my portion, you are responsible for the slides only, for the exam. However, I personally advocate, you are medical students, you have many years, in front, so try to go through the book also. But for the exam purposes, slides are okay. Also, over the years, I've seen students coming in, sitting for some time after doing their attendance, and then they go. See, I do not pressurize anybody to sit there. But if you don't want to attend, it's your wish. But at least if you want to leave, just do it at the beginning of the session. I'm not going to stop anybody. I only want those people who are interested in listening to what I have to say. Okay? And I'm not judging anybody here. All right? Also, if you see my slides, there is usually a diagram, an illustration given, and the text given. I hardly read the text. That text is for you. I can just put a diagram and talk about it, but I give you text for you, okay? And usually I repeat something which is difficult twice. Also, questions are encouraged, but it would depend how long my resource is. If it is longer, then I might take questions at the end. And if I have time, I'll answer them there and then. Okay? Other than that, tutorials are there. I will come for the tutorials. I encourage you to come there because I'll be discussing some sample questions also. And otherwise, you can come to my office anytime. If I am available and I have time, which I have most of the days, I will answer your questions. Okay? Any questions from your side before I start? All right. So let's go back. So without further ado, I know all of you are kind of stressed also. You have exam next week, but you know, we have to continue the process of giving you lectures because we are very short on time and we cannot give you a prep week. So bear with me, I'll go slow with this resource, and I know you can ask me questions, or maybe later on once you have gone through it properly, you can come again, okay? So let's talk about protein trafficking. So now I had already mentioned that proteins are synthesized in our cells. What's the organelle? Let's revise your exam content. What's the organelle that is synthesizing proteins? Ribosomes. Now they are synthesized by ribosomes, but we really need to know that those proteins that are made, how they get transported to different destinations. So this resource will cover those points. Now, as you mentioned that proteins are synthesized by ribosomes. What are the two types of ribosomes that you know of? So, as you mentioned, there are free ribosomes, meaning they are free in the cytosol, and there are ribosomes that are bound to the membrane. Mostly they are bound to the endoplasmic reticulum. So, they are called as bound ribosomes. Now, do you think the proteins that are made by free ribosomes and the proteins that are made by bound ribosomes, do they have same destinations or it is very random? Different, yes. So anything that is going inside an organelle, specifically any protein going to the nucleus inside, anything going to the mitochondria, or anything going to peroxisome is synthesized by free ribosomes. Even anything that is staying in the cytoplasm, it is synthesized by ribosomes that are free. On the other hand, proteins that are associated with membranes, specifically membrane of the ER, the Golgi complex, the plasma membrane, or any protein that is secreted outside of the cell is synthesized by ribosomes that are attached to the attached to the endoplasmic reticulum. Clear? Now let's look at trafficking or transportation of proteins that are synthesized by bound ribosomes. Again, just focus on the picture and listen to me as if I am telling you a story. Trust me, if you listen to me, you will not have a problem in memorizing the whole process. Okay? Now, if you see here, there are steps given 1 to 5, correct? Now, what happens? Initially, all ribosomes are free. Okay? Now, for example, in the first step, an mRNA comes and binds to a ribosome, a free ribosome. And then ribosome starts making a protein. So as it is making a protein, the N terminal is coming up. You have taken translation and you know that when the protein gets synthesized, the N terminal starts coming up. So the N terminal of the protein is coming up. It has a sequence which is called as signal sequence. Now when that signal sequence pops up, a protein is there in the cytosol which is called as SRP. This protein can recognize that signal sequence and this protein will go and bind to that signal sequence. So in the second step, if you see, CRP goes and binds to that signal sequence. Now CRP is a big protein, so it has the ability to drag the whole complex, the ribosome and the protein, towards the membrane of the ER, and it binds the whole of the complex to the receptor which is called as SRP receptor. This receptor is present near the opening in the ER membrane, and that opening is called as translocation complex. Okay? Translocation complex is simply a scientific term for an opening in the ER membrane. Now once the whole ribosome is bound to the membrane, the translation keeps on happening and the protein, it starts entering into the lumen of the ER through that opening. So if you see that the protein is entering inside the lumen, you see it? Once the translation of the protein is complete, the ribosome, it will get detached, it will become free again. The protein is now completely inside the lumen. The signal sequence, as you can see, it got removed. And this protein, which is inside the lumen of the ER, will now go through certain modification inside the ER. It can stay in the ER or it can get transferred to the Golgi complex. I repeat it again. So initially all ribosomes are free. Now when an mRNA comes and binds to a free ribosome, it starts making a protein. Now as the protein is made, what comes up on the N-terminal? Signal sequence. This signal sequence is recognized by a protein present in the cytosol.
صباح الخير يا جماعة. بخصوص الكويشنير تبع الاكريديتيشن، شفنا الريسبونس ريت من عندكم فكان تقريبا 50%، وهذا غير كافي. فضروري جدا اللي ما عباش الكويشنير سيرفي تبع الاكريديتيشن يعبيه as soon as possible. طبعا الفكرة انه الاكريديتيشن بالنسبة لكم اهم منا احنا، لانه حاليا مثلا في الجامعات الامريكية ما بيقبلوا اي طالب. بالنسبه لليو اس ام ال اي اللي بحب يروح يكمل في امريكا، ممنوع اصلا يدخل على امريكا يكمل بدون ما يكون جاي من جامعه اللي هي معتمده accredited. ف it is more important for your future than for us. فلازم تعبوه. هاي النقطه الثانيه بالنسبه للسكيل الموجود هو معكوس، يعني 1 هي satisfied و 4 not satisfied، بالعكس. فديروا بالكم لما تعبوا. if you are happy about the item you put 1. If you are not happy, you put 4. معكوس، تمام؟ We are looking forward to your cooperation on this. Very important for your future and for the institution as well. Thank you. Thank you, Deeba. Appreciate it.
All right. So then the SRP goes and binds to it and it drags the whole complex to bind to its receptor, which is called as SRP receptor. Where is that SRP receptor? It is located near the opening in the ER lumen. Now as the whole complex is bound to the membrane, the protein that is getting synthesized, it starts entering inside the lumen of the ER. And once the translation is over, the ribosomes get removed, the signal peptide gets removed, and the whole protein is in the ER. It can undergo modification, it can stay in the ER, or it can go to the Golgi complex. Clear? Now, as the protein is getting translated and entering inside the lumen of the ER, at the same time, modification of the protein also happens, or post-translational modification happens. That I think you took it yesterday, correct? So, since the modification and translation is happening side by side, it is called as co-translational process. Now, the most common type of modification that happens to a protein is adding sugars to it. What is it called? Glycosylation, correct. So, as the protein, now focus again on the figure, as the protein is entering into the lumen, in the membrane, there is a molecule present which is called as dolicol. It is a lipid molecule. Everything is mentioned in the text, okay? So, this dolicol molecule has a long chain of sugars attached to it, and that long chain of sugars is called as oligosaccharide. Oligo means many, saccharide means sugars. So, what happens when the protein has come inside, fairly inside, these sugar chains, they get released from the dolicol and they get attached to the protein. Okay. Now again question, do you think these sugars can be attached randomly anywhere on the protein, or is there a specific sequence for it? See, nothing in nature happens randomly. There is a perfect order in each process that is done. So here also these sugars are added to a particular sequence that is present in the protein. Proteins are made up of what? Amino acids. So there is a sequence of amino acid where the sugars attach, and that sequence is SNX SIR or SNX THR, sorry. X indicates that in the middle it can be any amino acid, but the sugar gets attached to the SN amino acid. I am not giving you full forms here because this is something you will take in biochemistry in the next semester. So just remember SN. So the whole sugar moiety, it gets attached to the SN of the sequence, but the sequence has to be so, SNX THR or SNX SIR. And since it is attached to the amino group on the SN, this type of glycosylation is called as N-glycosylation. Another glycosylation takes place, but that takes place in the Golgi, which is called as O-linked glycosylation, in which the same types of sugars are added to the same sequence, but on the SIR and THR part of it, okay? And since they are added to the hydroxyl group of SIR or THR, it is called as O-linked glycosylation. But that happens in Golgi. So understood? As the protein is coming in, glycosylation is also taking part. Which molecule is holding the sugars? No, no. On the membrane, what is the molecule that is holding the chain of sugars? Dolicol. So dolicol transfers the sugars to which amino acid? SN. And if it is getting transferred to SN, it is called as N-glycosylation. However, in Golgi also same glycosylation happens, but the sugars are transferred to the SIR and THR, and that is called as O-linked glycosylation. Understood? All right. Let's talk about what happens once the protein has been modified in the ER. Now, if you go, mentally through, I'm sure you are prepared by now. So, after the endoplasmic reticulum, which is the next organelle that comes into picture? Golgi complex, correct? And enlighten me, what is the structure of the Golgi? It has different parts, no? Yes? And something in the middle. No, all of them are cristae, you are correct. It has a cis, it has a medial, and it has a trans, correct? So, you only studied two, okay, fair enough, but there is medial also in the middle. And that also depends on how big the stack is. Now, what happens, the proteins that have been modified in the ER, they can stay in the ER and function in the ER membrane, or they can get transferred into Golgi. Now, for the transferring of the proteins to Golgi, what happens is that the membrane of the ER, it bulges and it forms a vesicle around the protein that needs to be transported. Now, when that vesicle has been formed, it moves and it goes and it fuses with the cis portion of the Golgi, and the protein gets inserted into the Golgi. Okay? Now again here, do you think the vesicle that is coming from the ER, it can randomly fuse anywhere on the cis Golgi? No, it cannot. Why? Because if it will do so, then that protein might miss certain kind of modification that it needs to go through. Okay? So how it is ensured that it gets fused at the correct position? So what happens, the vesicle, let's focus on the second one. The vesicle that is coming from the ER, it gets coated with a protein which is called as V-SNAR or vesicle SNAR. Okay? And on the Golgi, the area where that protein should come, that area is coated with another protein which is called as T-SNAR or target SNAR. So these are two tags. One tag is on the vesicle which is called as V-SNAR, and one tag is on the Golgi which is T-SNAR. Simple? So I don't think so I need to repeat this. This is fairly understood. Now let's see what happens in the Golgi. Now the protein comes to the Golgi, and you know that Golgi is the sorting organelle or the post office of the cell, where all the proteins come, they get sorted and they go to different destinations. So what happens, the protein gets inserted into the cis, and all the stacks or the cristae of cis and medial, they are filled with enzymes, even trans, they are filled with enzymes which bring about modifications in the proteins that are passing through it. And modification happens depending on what is the destination of the protein. For example, if the protein has to work in the lysosome, so it needs phosphorylation on the mannose sugar. Phosphorylation means adding a phosphate. So any protein that is supposed to be part of lysosome, it will get a phosphate on the mannose sugar that is attached to that protein. And now the Golgi would know, oh, it has mannose 6-phosphate tag on it. This means it has to go to lysosome. So it is like an email address on that protein, that okay, it needs to go to that destination. Any protein that needs to go outside the cell, it will have a different tag on it. Okay? And of course, I already have mentioned, I am not going to repeat it again. Other type of modification that happens in Golgi is O-linked glycosylation. Now, once modifications have happened, the protein reaches the trans Golgi, where the final sorting happens for each protein. So, if this is the trans Golgi, the upper part of the desktop, the proteins that are going to lysosome will be in one area. The proteins that are going to the plasma membrane are in another area. The proteins that are going outside the cell, they are in the third area. So, they get sorted and then vesicles get formed around them and they go to their own destinations. Okay? Any questions until here? No. Oh, this is just an animation to show you that how protein from the ER, it gets transported to the Golgi, and as you see, even from within the Golgi, a vesicle has to bud out and then it has to fuse again. A vesicle has to bud out and fuse again. And the same coating has to come. So, a V star will come, a T star, wherever a T star is, it will fuse. Okay? Now, let's talk about how lysosomes get formed, or the proteins that belong to the lysosome, how do they reach lysosome. So, what are lysosomes, by the way? Organelle, okay. It is filled with degradative enzymes. What is the pH status of the? So, it is acidic, correct? So, it has acidic pH, and as you correctly say, it has degradative enzymes which are called as acid hydrolases enzymes. Acid, why? Because they function in an acidic environment. Okay? Now, let me tell you the story. Again, focus on the black picture that is given here. So, let me start the story from the ER. So, a ribosome with SRP will come and bind to the ER, a protein will get synthesized. In case of lysosome, it is acid hydrolase enzyme. It gets synthesized. In the ER, it will get a mannose on it. Okay? Now, from the ER, acid hydrolase enzyme with a mannose on it, it will come and it will fuse with the Golgi, as you can see here. Now, as the enzyme is traveling through the Golgi, it gets a phosphate on it. The red dot that you are seeing here is a phosphate. And where is it getting the phosphate? It is getting the phosphate on the mannose. So, the tag becomes mannose 6-phosphate. So, all the acid hydrolases, they will have mannose 6-phosphate, and then finally they will reach the trans Golgi. Now, in the trans Golgi, there are receptors present, as you can see here. These receptors, they do not recognize acid hydrolases enzyme, but they do recognize the mannose 6-phosphate tag on them. So, any hydrolase enzyme which does not have this tag, it will not get transported. So, all the acid hydrolases with the tag, they will bind to the receptors in a particular area, not randomly. If you see, they are only bound to a particular area. Now, once they are bound, the membrane, it bulges out and it forms a vesicle. And that vesicle is coated with a protein, and that protein is called as clathrin. It is mentioned in the text. Now, clathrin is a protein which is very important in helping in making this vesicle. If clathrin protein is not present, this vesicle containing the enzymes will not get made. Now, once the vesicle has been formed, the clathrin coating, it gets removed. So the only function of clathrin here is to help in making the transport vesicle around those enzymes. Now as the vesicle has been formed, as you can see here, it is filled with enzymes attached to the receptor and it is moving towards the cell membrane. At the same time, whatever proteins that have to come from, say, outside of the cell or the plasma membrane, they come inside the cell and engulfed in a vesicle which is called as an endosome. So a vesicle carrying the enzyme coming from one side and the proteins from outside of the cell, they are coming from the other side as an endosome. They come and they fuse. And when they fuse, they form the primary lysosome. Have you heard of primary and secondary lysosomes? Okay. So simply, what are primary lysosomes? Primary lysosomes are when the vesicle carrying the enzymes and a vesicle carrying any protein that needs degradation, they fuse together. That's primary lysosome. Now, do these enzymes become functional immediately? No. Why? Because they are still attached to the receptor and they cannot function unless they get released from the receptor. Now, for releasing them from the receptor, hydrogen ions are needed inside the cell, inside the lysosome. Do you remember, again this is revision, do you remember which pump puts the protons inside the lysosome? V pump, proton pump or V pump. That V pump, it pumps hydrogen enzymes, as you can see here, from the cytosol inside the lysosome. And as the protons are increasing and increasing, the pH is becoming acidic and acidic. Now when the pH becomes acidic, the enzymes, they get released from the receptor. The receptor go back to the Golgi. The phosphate gets removed and the enzyme is now functional. Okay? And when the enzyme is functional and breaking the protein down, it is called as secondary lysosome. This is not mentioned in the slide, primary and secondary. And I am not going to ask you what is primary and what is secondary. It was just additional info. Okay? Very quickly, I'll just repeat this and I want interaction from you. So acid hydrolases are the core proteins in lysosome, correct? So a ribosome binds to the ER, it produces acid hydrolase. What gets added to the acid hydrolase in the ER? No, in the ER, mannose, sugar gets added, okay? Now acid hydrolases with the sugar, they enter into the Golgi. In the Golgi, what gets added? Phosphate on the mannose. So it becomes mannose 6 phosphate. This protein that now gets transferred to the trans portion of the Golgi and it binds to the receptor which identifies the whole acid hydrolase. Correct? No. It only recognizes the tag. Remember, trans Golgi receptors will only remember the email address on the protein. And the email address on acid hydrolases is mannose 6 phosphate. So it will only recognize mannose 6 phosphate. Once the acid hydrolases are bound to the receptor, a vesicle gets formed. Which protein is playing a role in forming the vesicle? Clathrin. Once the vesicle gets formed, clathrin gets removed. The vesicle carrying these receptor bound acid hydrolases, at the same time any protein from outside of the cell or any old organelle inside the cell, it gets enclosed in a vesicle and they combine. When they combine, it is called as a primary lysosome because still the enzymes are not functional. Now what happens? A proton pump or a V pump, it starts pumping protons inside this lysosome As a result, what happens to the pH? It becomes acidic. Now, when the pH becomes acidic, the acid hydrolases, they get released from the receptors. Receptors go back. The phosphate also gets removed. Mannose stays with the enzyme. Okay. The phosphate gets removed and the now enzymes are functional and they will start breaking whatever they need to break. Okay. And such lysosomes are secondary lysosome. Clear to everybody now? Secretion of the proteins from the cell. Now, by now you know that any protein that needs to go outside of the cell has to be made by a ribosome that's attached to the ER, because then it will go from ER to Golgi and then outside. So, regarding the secretion of the protein outside of the cell, there are two important processes that control that secretion, and those processes are called as constitutive and regulated. Now, constitutive secretion is for those proteins that are constantly needed by the outside of the cell, like any protein which is part of the extracellular matrix or serum albumin, which is an important part of our blood. So, for secretion pathway, or structural means constitutive only. So, do not get confused. Structural and constitutive means the same thing. Now, as I mentioned, this secretion happens for proteins that are needed almost all the time. And since it is needed all the time, no external stimulus is needed on the cell. It happens automatically, okay? And usually it happens for proteins which are called as housekeeping proteins, which are taking care of the outside environment of the cell, alright? Now then there are regulated secretion. Now this happens for proteins which are needed only when there is a stimulus. Very good. So, for example, for digestive enzymes or hormones. Now, let me give you a very simple example when we are eating something. All of you know that when we eat, there is secretion of saliva, and saliva has certain enzymes. Yes? So, when we are eating and chewing, that chewing becomes a stimulus for the salivary glands or the salivary cells. Automatically they will start secreting enzymes in our saliva which starts acting on that food. So that chewing process is a stimulus. Otherwise, if those enzymes are secreted in our mouth without any food, they are so strong that they can harm our tongues, our cheek, or any anatomy of our mouth. So without stimulus, if they get secreted, it's not good for our mouth anatomy. Okay? So you got the picture? Regulated is only when a stimulus is there. Which hormone is secreted when our blood sugar is high? Insulin. So is insulin secreted when we are in fasting state? No. So what is the stimulus for secretion of insulin? When blood sugar levels are high. So that acts as a regulatory secretion pathway. Clear to everybody? I will stop here. Okay. I'll meet you again tomorrow. If you have any questions, you are welcome to ask. Otherwise, you can take your own time. It's not a problem. Okay. Thank you for your attention.